It is my pleasure to introduce today's speaker, Andrew Thurber. Andrew did his undergraduate work at Hawaii Pacific University, where he studied marine biology, majored in marine biology and math. Worked with a guy that I went to grad school with, Frank Smith. Then he went to Moss Landing Marine Lab, first place I had a paying job as a marine biologist. And Moss Landing does almost as good a job making master's students as we do, except that they do it really slowly. So Moss Landing is famous for having the longest time for people to get a master's degree. The record is apparently 12 years, and the average is apparently 6 years. And Andrew did it really fast. He did it in 4 years. But Moss Landing is also very good at getting people into good graduate schools. And so he went from Moss Landing to Scripps Institute of Oceanography, where he worked with Lisa Levin on deep sea biology and methane seeps, and that's the sort of stuff he's going to talk about today. And now he's doing a postdoc at OSU, working through polar programs, which is funded by NSF. Okay?
Well, thank you very much for the introduction, and thanks for having me here. It's always nice to have everybody in a good mood by getting money at the beginning of the talk. There's a lot of wonderful questions in science, and many of them are really not new, and yet we still don't have answers to them. And one of those questions, I think, was very nicely and succinctly put in the very first issue of Nature in 1870 by Sir Wyville Thomson, when he said, "There's little or no life in the depths, and naturally there are no plants, but the bottom of the sea is a mass of animal life. On what do these animals feed?" That question still remains a question in science, and we're taking many different approaches to answer that. But as we were essentially trying to just figure out how the deep sea works, a couple of discoveries were made which were unexpected. The first one of these was in 1977, the discovery of a hydrothermal vent off the Galapagos, and the second one was the discovery of a methane seep just a few years later in 1984 off the Florida escarpment. Since then, these originally thought to be extreme and unique habitats have been found all over the world. All the red dots are hydrothermal vents, all the blue dots are methane seeps, and essentially this map, as soon as you make one, is out of date because the more we look, the more we find. What you can see is there's essentially a network going across the globe. And so I'm going to tell you two little stories that I've been involved with over the past 5 years or so of what we found when we started to look at what things eat at these different habitats. I'm going to start by telling you about a symbiont-bearing animal, specifically a Yeti crab we discovered off of Costa Rica at about 1,000 meters depth. And the second one we also found off Costa Rica at about 1,000 meters depth. And this is not a symbiotic relationship, but instead it's a heterotrophic relationship of an animal that eats a domain of life, which to this point has essentially not been involved in any sort of trophic or food web studies. So even many of these habitats, after they've been discovered, we discover more things about them when we return. In fact, this site had been studied numerous times, had been visited numerous times, and yet strangely, when the subpilot took a left instead of a right, he stumbled across this. This is essentially a species being discovered. This is the video off the sub. And the subpilot looked over and saw this group of crabs sitting there rhythmically waving their arms or kilo heads, if you want to be proper, back and forth in areas where there's sea fluid coming out of the seafloor. And we know that because we found them on Bathymodeal mussels, in pits within carbonate rocks, on the mellibrake tubehorn bushes, which you can see in the background, and they just sat there rhythmically waving their arms back and forth. This is what they look like when you see them essentially dead and many months later in the lab. But what was special about them is that all their hairs that cover their entire body were also covered by bacteria. And this was even more surprising because earlier that year, so this was in 2006 when we first discovered them, the very first Yeti crab was found. This was found in a hydrothermal vent much farther south and much deeper than these animals and did none of the arm waving motion, but it really captured the imagination of the world. It's been on more National Geographic photos than I can talk about. But this entire species was known from one specimen, and it was so unique, it created its own superfamily, which is still a very robust family, and this was the second species that would fall within that group, or at least we thought from its overall structure. It turns out that's most definitely true. Instead of getting one individual to define the entire species, family, superfamily, we collected quite a few of this methane seep Yeti crab and were able to figure out many of the questions about its biology that we were not able to do or the scientists who discovered the original Yeti crab were not able to do with that specimen. And so we just kind of wanted to know what it does. What does it eat? Why is it farming these epibiotic bacteria, or essentially using these bacteria that are growing on it as a source of food? And then where does this species and then the symbionts, or essentially the bacteria that grow on it, how can we try to understand the global biogeography of methane seep and hydrothermal vents? Because remember, this is the first hydrothermal vent individual that had epibiotic bacteria growing on it that belongs to the crustacean and has received study. And really, why is it dancing, as I like to call it? Why is it waving its chelipeds back and forth? I'm essentially going to answer these questions through a variety of different ways. First, if it is getting energy from the seep, it is getting energy from its symbionts, then it has to get energy from chemosynthetic food sources rather than photosynthetic food sources. Secondly, if it is one of the kind of missing links for these other species, is you need to be able to trace those bacteria into its diet in a form other than just knowing it gets chemosynthetic energy, and lipids provide a lot of technique, or essentially a specific technique to allow that. And then what's remained quite enigmatic for the other groups that have epibiotic bacteria is they lack a mechanism for which to harvest bacteria. So even if they have bacteria growing all over them, they're not able to harvest this. And this is really limited to the shrimp, the crabs, and barnacles to a lesser extent. But none of these have essentially been found to eat epibiotic bacteria that have them. And then finally, it would be great if we can actually demonstrate that they are able to eat these bacteria. And so the way we're going to start doing that is we can use the carbon isotopic ratio, really bright, to figure out whether or not they're eating chemosynthetic versus photosynthetic food sources. This simply takes advantage of the fact that the carbon comes in two stable isotopes, 12 and 13, and the ratio of carbon 12 to carbon 13, when you compare to a standard, lets you talk about the delta 13C. This is simply a way to have comparable carbon 12 to carbon 13C comparable across many studies. Photosynthetic enzymes select for a certain ratio of carbon 12 to carbon 13, and it really lives right around in this minus 15 to minus 24 per mil is the notation for carbon. Chemosynthetic food sources are far skewed to this, much favoring carbon 12. And so you can see anything that doesn't fall within this range of photosynthetic production on the carbon axis, which is here, is getting energy from methane seeps. You can see the black dots, which are the actual muscle tissues from this crab, don't fall within that green area, so we know they're not eating photosynthetic production. Instead, they're getting energy from the methane seep that they live. You can learn a lot from nitrogen. I'm not going to talk about it, so if you know what that is, you can look at that and think about it. But these green dots, I should say, the photosynthetic values were collected from the water column right over the methane seep, as well as all up and down the Costa Rican margin where we collect these animals to try to get a good kind of wide-ranging view of what photosynthetic production may look like. And it's very clear that the Yeti crab was not getting energy from photosynthetic production. Now, lipids are a very similar thing because we get our carbon from what we eat. We also get many of the lipids that form our cellular membranes from what we eat. And this used to be, pretty hard to think about. But now if you go into the cereal aisle and you look up and down, there's all these, have PUFAs, DHA, EPA is listed on all the different cereals, saying it helps with brain and, success of childhood or children. And that's because we get essentially the fatty acids that form our cellular membranes also from our food. This is, for example, what a fatty acid looks like. It's simply a carbon chain. They're named by the number of carbons within the chain and then the number of double bonds they have. For example, this is a 16 carbon chain fatty acid and has no double bonds. And why we need to get fatty acids from our food is that us, as well as most metazoans, can only synthesize certain fatty acids. In fact, we're very limited compared to bacteria or photosynthetic producers. And we have a single elongase, so we can add 2 carbons to it. So we can take we can create a 16-0 fatty acid. It's the only one we can create kind of de novo. We can add 2 carbons to it, only right here, using elongases. Or we can add a single double bond, 7 carbons from the terminal end. So that's all we can do. And by knowing what we can do, and all metazoans can do, we can come up with a suite of all the fatty acids that we can create and we can synthesize. So this is essentially everything, if we were just eating something that provided none of those nutrients, what we would expect to find within our cellular constituents. However, we get our fatty acids also from our food sources because it's energetically favorable not to synthesize all those. And so by looking at the relative proportion of fatty acids from two different food sources, for example, this, the epibiotic bacteria on the Yeti crab, and lots of 16-1N-7, compare that to the photosynthetic fatty acids, which are abundant. For example, this 22 carbon, 6 double bond, the first one 3 from the end, and see which one's more prevalent in the muscle tissue, you know whether that animal is eating more of the bacteria or more of the phytoplankton. So this is how we can essentially interpret these different fatty acid signatures. On the y-axis, I have the percent of the total fatty acids that were found within the phytoplankton in green, essentially the bacteria covered with seedy tips. So we essentially just cut this little tufty part of lots of the seedy or hairs off to get an idea of what the bacterial fatty acid signature was. And then we also took the muscle tissue inside. The muscle tissue is a time-integrated view of its diet. And then these are all the different fatty acids, which form more than 1% of any one of these sources. First off, what you can see is that the blue and the black, the muscle tissue and the bacteria, share many similar patterns of abundance. So 16-2, this branched fatty acid, that's what the eye indicates, is only present in these two food sources. But we can use this a little bit more specifically if we use some knowledge about what other, so for example, phytoplankton are very abundant in 25N-3, 22,6, N-3. These are what we try to get from our cereal. And we find that, as we would expect, they were very abundant in our phytoplankton food source. The rest of these are actually mostly synthesized by anything, including us, except for one, and that's the 16-2 fatty acid. The 16-2 fatty acid is really only abundant in other crustaceans that have epibiotic bacteria, specifically Rhinoceros hexaculata, or the vent shrimp found in the Mid-Atlantic Ridge, or Shingai crossneri, a crab that's found off of Japan. It's also found to a lesser extent in certain mussels within the Bathymoleolin group, but quite rarely. And of all of the samples that I've collected at Costa Rica, as well as a variety of different methane seeps, I have not seen the 16-2. So it's a very unique fatty acid. And what that means is that by knowing that it's in the bacteria, we can see whether or not it's actually taken up and consumed in the tissue. So let's say there's I confused myself, which is never a good sign when you're giving a talk. So there's only one real source that we know of for the 16-2 fatty acid. It's the epibiotic bacteria that grow on this crab, and as kind of a nail in the coffin to show that it is consuming those bacteria, that fatty acid was also abundant and present within its tissues. So this is a very powerful way to see that it is actually consuming the epibiotic bacteria that grows upon it. Now, that's not overly unique. So Rhinoceros has been shown to be able to trace all these fatty acids in isotopic approaches, which I've just discussed. However, what it wasn't able to show is we don't know how it might actually be able to harvest these bacteria, because it doesn't have the morphological adaptations to be able to consume the bacteria that grows within its carapace. And so we were surprised, and actually I should say delighted more than surprised, to start looking carefully at all the different seedy, and it turns out there's 3 different kinds of hairs that grow on this crab. There's stout, potentially protective seedy that grow on the back of its carapace and on its chelipeds. And then there's also whip-like seedy, which is where most of the actual bacteria grow. And then there's this third type, which we call comber seedy, which only grow on 2 of its mouth arms, its third maxilliped and its second maxilliped. Here's where the third maxilliped lives, and then right behind that is the second maxilliped. If you look closely, the tips of these seedy are completely dominated by combs. Now this is an scanning electron micrograph of it, and you can see that these combs are very defined and potentially could be used to scrape the bacteria off of the whip-like seedy, which have all the bacteria. Here's a picture of the whip-like seedy with epibiotic bacteria growing. When we actually started looking at these more and more under the microscope, we found it was very common to actually see the epibiotic bacteria morphotype stuck within the combs as a way that they can harvest the bacteria and move it towards their mouth. And so they have the morphological adaptation to be able to harvest it, but do they actually do it? So we pulled up a crab from 1,000 meters, and we put it in an aquarium, and we essentially just let it sit. And this is what it's doing. Here's its third maxilliped, those are its second maxillipeds, and it spent a lot of time simply using these comber seedy to scrape the bacteria off of the whip-like barb seedy, move it up towards its mouth, which would then scrape its third maxilliped with its second maxilliped, and put those bacteria kind of straight into its mouth. So we're able to show essentially the entire cycle of that they're getting energy from chemosynthetic food sources, that they have a unique fatty acid which we can trace from those bacteria into its muscle tissue, showing that it's an important member of its diet. And then it has the morphology and actually the behavior to be able to harvest these. We even decided to go one step further. We dissected very few specimens. All the other specimens are type specimens in different museums around the world, so we didn't want to just go cut them up willy-nilly. But if you look at the very front of its mouth, you could find the amorphous species of bacteria complete. On the other side of the cardiac stomach, you find chopped up little pieces of these epibiotic bacteria. So we essentially were even able to follow it into its gut. You'll notice that there's a lot of other stuff in its gut. Other stuff, or detritus is I think the common term for other stuff when you get to the deep sea. And if you look carefully on the whip-like Corvallis, for example, this one, you see there's a whole lot of other stuff in it. There's tons of detritus. And so you also, if you look back, you also, or think back, there was also a few photosynthetic biomarkers that were present within both the epibiotic bacteria samples as well as the food sources. We think that as the animals are waving their arms back and forth, they might actually pick up different sources of food, for example, phytoplankton that's raining down, to augment their diet and provide additional nutrients that they wouldn't get from simply a bacterial food source. One thing to think about that, though, is these have had quite a rocky ride up from 1,000 meters in a sub-basket shaped around with a bunch of rocks. And so it's not necessarily definitely happening in the deep sea because it might just be an artifact of the collection technique. And that's why they get covered in gunk, and then that's when we see them. But the question is, why are they waving their arms? Symbiont-bearing organisms at seeps and vents have a variety of different techniques to try to increase the productivity of their symbionts. A classic one is clams pump oxygen from the overlying water column to be able to suck up the sulfide from their foot, have chemosynthetic reactions by the bacteria that are symbionts within them to create energy. I think the most circuitous but really interesting story right now is these tube worms which leak sulfate out of their roots, which the sulfate then reacts through a microbial mediated process with methane, increasing hydrogen sulfide. The hydrogen sulfide is what they need for energy when they combine with oxygen in their plume. And so simply by leaking sulfide, it's a mechanism to increase the by leaking sulfate, they're increasing the sulfide, which can then increase the overall productivity of their symbionts. And both of these are ways to essentially increase the amount of solutes that their symbionts get access to. And one of the problems is if you have something, whether it be a photosynthetic macroalgae or phytoplankton, you essentially are constantly sucking CO2 and everything you need out of the water column. With no water movement, it becomes stagnant, you run out, and create a boundary layer. You run out of solutes. So for example, if you have the distance from an arm and you're a Yeti crab, if you just hold your arms in one place, you're going to run out of both the oxygen and hydrogen sulfide, potentially methane that you need. But by waving your arm, you get rid of those boundary layers, increasing the overall productivity of your symbionts and allowing you to grow more rapidly. So this is the first one that we're able to kind of complete the story about actually eating epibiotic bacteria when you compare the different crustaceans from hydrothermal vents around the world. But it also is the first methane seep one that's undergone study in this regard. And so what this is the phylogeny based on 16 S-ribosomal RNA of the epibiotic bacteria. And there were two main groups that came out. The epsilon proteobacteria, these are commonly sulfide oxidizing bacteria, at least on shrimp, as well as gamma proteobacteria, which can either be sulfide oxidizers or methanotrophs in different systems. And when we took our methane seep bacterial data and we threw it in GenBank and compared everything that's ever been sequenced, we found that without question, they were all most similar to those three other species of crustaceans from hydrothermal vents that have epibiotic bacteria. So here we have our Yeti crab, the methane seep Yeti crab in green. We have Kiwa hirsuta. This was the original Yeti crab from a hydrothermal vent collected from south of the equator. Shinkai crossharai, which is from Japan, and Rhinoceros exoplata from the mid-Atlantic ridge. And this is a very poorly kind of constrained tree because it was constant small deviations in the genetic code, which means there's no clear pattern or trend, which is what you would expect if there was a lot of mixing or small-scale evolution that went on rather than clear lineages. So this suggests that, first off, from everything we've sequenced, there's some sort of weird global distributed methane seep and hydrothermal bacteria which specialize on living on crustaceans, essentially shrimp and crabs. And that essentially is at least two different clades or two different fire level distinctions, the gamma-fruited bacteria and epsilon-fruited bacteria. And if you think about that in a biogeographical sense, this is where the original Yeti crab was collected, this is where Cuba curvida was collected, and then this is the Shinkai Krasnorai, and this is where the Mid-Atlantic Bridge molecular sequence. There has to be some sort of really incredible connectivity among all of these sites that's working at an evolutionary time scale where there's no real pattern within the tree, even though the hosts are very phylogenetically different. And although this was clear from just looking at an initial pattern was found looking at the hydrothermal signals, by adding in methane seeps to a connectivity matrix, especially if you're trying to think of bacteria on the or dispersal on a bacterial size scale, it really is worthwhile if you have many more stepping stones along. By adding in methane seeps, all those known and unknown, it makes it much more kind of plausible to see how bacteria may independently from a host connect and create this deep sea specialized group that only lives on epibiotically on crustaceans. I think it's a really intriguing idea. The more we discover, the more we'll find out if this happens to be true, especially the more we sequence non-charismatic megafauna that dance around in the deep sea. So essentially the first part of the talk, epibiotic bacteria, the main food source for this animal. It has a unique dancing behavior, which I think increases its epibiotic bacterial's productivity, allowing it to thrive at these one habitats. And also, it's neat because it adds methane seeps into our understanding of biogeography of bacteria within reducing habitats that only live on crustaceans. Now I'm going to change a bit. And the reason is symbiont-bearing animals have been very useful and taught us a whole lot about the deep sea. But if we think about biogeochemical cycling and the role that trophic interactions can have, the role of heterotrophic interactions, I think, could be argued to be more important than those symbiont-bearing ones. And so we're going to switch to a heterotrophic organism. Now, as somebody who studies trophic interactions, I think they're the most important interactions on the planet. But it's good to demonstrate that to people who don't study trophic interactions. And so an easy way to look at that is if we think of a simplistic, oversimplified limnologic or lake system. We have a simple phytoplankton and one grazer. There can be such heavy top-down forcing or grazing pressure that you have a beautiful, nice, clear lake. If you introduce a fish, very unlikely to be this particular species of fish, you can then have the fish exert such a top-down force on the grazer, for example, as that, and then the phytoplankton are able to bloom, and you get a beautiful green lake. Does anybody know what the fish is? Orange rocket. The problem with being a deep sea biologist and talking about limnologic systems is I have no pictures of trout. And so that deals with phytoplankton, but bacteria, the more we study, the more we realize that bacteria are incredibly important in many biogeochemical processes, from nitrogen to oxygen, everything really involve bacteria in some way or another. And this is, again, a new idea. Lindemann, 1942, landmark work, created a lake food web. Right at the center of it, he put bacteria and ooze. That was in 1942. Here's a wonderful paper, and I don't mean to insult this paper. It's a great read about some of the most cutting-edge ways to look at food webs in 2009. There's been no change other than bacteria are not quite in the center anymore, but still just called bacteria. And one of the greatest diversities we found on the planet is that bacteria are incredibly diverse and do many different things. In fact, in 1977, they found out that everything that we were calling bacteria are actually two completely different lineages, the archaea and the bacteria. While bacteria are incorporated into some sub-food web studies, archaea are still completely left out. And this is a mistake because these archaea, this entire domain of life, we now know may be the most, or ties for the most abundant form of life on the entire planet due to their ubiquity in the deep biosphere. They perform many important processes, including methane oxidation, methanogenesis. The more we study, we find out they're very involved with nitrogen cycling and may be important mitigation of waste. Not to mention they're the underlying cause of much of the chemosynthetic production at methane seep systems. However, we don't know if they're actually a potential food source because it's never been tried. So this next kind of path was to figure out, are archaea a viable food source? Can metazoans even consume them and get energy from them? It's a very basic question we don't know the answer to. Secondly, if they are consumed, is there any sort of smoking gun or specific biomarker way that we can identify any organism that's eating archaea? And thirdly, does archivory occur? Archivory is a word I made up to mean an animal that consumes archaea. So one of the most important, especially for somebody who studies methane seeps, reactions that archaea mediate is the anaerobic oxidation of methane. Methane is a greenhouse gas, 23 to 27 times more efficient at warming our atmosphere than CO2. It exists in vast reservoirs underneath the continental margin, and where it's released, the majority of it is consumed by a syntrophic partnership between a bacteria, essentially the candy coating of this, and bacteria, or an archaea, which is the red. They consume somewhere, but depending on what model you want to look at, between 60 and 90% of the methane before it can even escape the sediment, the seafloor, keeping it out of the atmosphere. It's kind of like putting a pin in a grenade as it comes up. So they also co-occur with metazoans, and it's a specific group of metazoans, the Dorvallated polychaetes. This is a picture of many of the Dorvallated polychaetes. The disadvantage of working on Dorvallated polychaetes is they hate being brought to the surface, and they tend to pop. So while these may be beautiful in situ, they don't make great microscope pictures. But if we go back to trying to identify animals that are going to consume them, we can again look at the relative ratio of carbon-12 to carbon-13. This is now on the y-axis. And on the x-axis, I have a variety of different food types. These are literature values. And because this entire system is based on methane, we can take advantage of the very skewed ratio of carbon-12 to carbon-13 that methane provides to look essentially for animals that may consume these anaerobic methane oxidizing archaeal aggregates. I should say the archaea take the methane, the bacteria are sulfate reducers, and they take the sulfate. There's some sort of unknown reaction, which we still don't know, that creates hydrogen sulfide, as well as energy, and also increases the alkalinity of the sediment, which I'll come back to in a second. But to go here, if you look at the carbon ratio of the biomass of the archaea, it has a relative ratio of minus 30 to about minus 101. The sulfate-reducing bacteria are slightly heavier, but still far removed from that photosynthetic values, which are all around here. The majority of them are much more light. Light meaning more 12, more negative. And then their lipids are even more selective for carbon-12, and skews this entire ratio down to about the heaviest archaeal lipid from an anaerobic methane oxidizing archaea is minus 60. That goes down to about minus 130. And sulfate-reducing lipids, so the bacterial fatty acids, are also quite light, although not quite as light. So it's an important system to study because it's the methane cycle, which we really don't have as good of a handle on as we would like. And secondly, it's a place where you have incredibly high abundances of archaea juxtaposed with a diversity of different metazoans that co-occur in sediment. But there was that initial question, can an animal actually consume, persist, and live on archaea? So I went to the lab, and I took this lab rat, Ophryotropa lebronica, from a Venice lagoon. I didn't actually take it, I just got a sample of it, and raised it on 2 food sources from each domain of life in antibiotic-laden seawater, so they wouldn't eat anything else. And found out that this particular worm, which is within the same family as those that we find at deep-sea methane seeps all over the world, was able to grow on all food sources equally. In fact, it potentially even grew better on archaea, as indicated in red here. This is the growth rate over 45 days. And bacteria than food sources, which have been raised on the lab since the 1950s. Oryza is rice, spinach is Spinacia is spinach. And then these are 2 different kinds of bacteria, 2 different kinds of archaea. So they can definitely grow on archaea. In fact, there's about equal reproductive success independent of food source. So it's a viable food source. Now, to tie back into the lipids, here's what we talked about, all metazoans being able to synthesize. And then here's 2 unique fatty acids, which are very abundant within the sulfate-reducing bacteria, which form half this syntrophic partnership that performs the anaerobic oxidation of methane. So using the previously discussed technique, if you have a sulfate-reducing bacterial membrane and you have a worm that eats it, it's going to take all of the de novo synthesized fatty acids that it can, indicated in blue, and mix it with those that it's getting from its diet in green to create its fatty acid profile. Now, a problem with archaea is they don't actually have fatty acids, so we don't know if any sort of fatty acid approach can work. Instead, they've got repeating isoprene units of a variety of different lengths and size, and they even have ether linkage instead of ester linkages to create the carbon backbone. But it would be an incredibly powerful technique if these very unique non-fatty acids, the lipids, were incorporated into the metazoan, and so we could just essentially look for them in the diet. So we were hoping we would find this to be the smoking gun. We could go look in every environment to see what eats archaea, and we found out that that did not work at all. In fact, they broke all of them down, even with very carefully controlled experiments and looking in situ, they did not uptake any archaea lipids. Even more confusing, this is a multidimensional scaling plot. All you need to know to interpret is that 2 points more similar or closer to each other are more similar than 2 points farther apart. And each one of these points is representative of the entire fatty acid profile of a worm fed one of the food sources. So for example, the diamonds or the spinach fed, and the gray or the eukaryotes, the black or the bacteria, and the clear or the archaeal food sources. And there's no pattern here. There's no statistical pattern here, there's no visual pattern here, that's because there's no pattern here. And the reason is because almost all of the fatty acids that this worm created were those that it can de novo synthesize. Not only was it not de novo synthesizing almost all of its fatty acids, if not all of its fatty acids, it was de novo synthesizing all the fatty acids that we don't think metazoans are physically capable of synthesizing. So that caused a bit of a problem, but it turned out to be a very useful technique in the end, because it allowed us to know that not everything is as you would expect from the literature. But then it brings us to kind of a challenging point, because we know archaea food source, we know that archaeal biomarkers are not necessarily incorporated, but how can we tell if archivory actually occurs? So now we're going to go to a unique environment to methane seeps. It's the inside of authigenic carbonates. Because remember, I talked about alkalinity being increased in sediment, and that actually causes the precipitation of calcium carbonate, which can form huge rocks. This is a picture off our coast here at Hydrate Ridge. This is authigenic carbonate precipitated by the anaerobic oxidation of methane. If you like fish, you can see that fish use it as a habitat. And although there's been a lot of animals that grow on it, sometimes very classic, filter feeders and things you see from seamounts, we decided to look inside the rock instead. And we found one particular worm. It's a new species of Dorvillia polychaete, which does fall within the Dorvilliid family, as well as a ton of extra large ambi-aggregates. This is a micrograph of that centropic partnership between bacteria and archaea. Now, this isn't the worm. We essentially cut this rock in half, peeled it apart, and we found both this metazoan population as well as this anaerobic methane oxidizing centropic partnership. And so, building on the previous figure that I showed you, here's the literature values of these anaerobic methane oxidizing archaea, the red or the amni or the archaea themselves, the green or the sulfate-reducing bacteria. And we're able to measure the isotopic composition of one of the abundant archaeal lipids within the sediment, quercetine. Quercetine is going to try to extrapolate to all the archaeal lipid values. We know that it's never more than 12 per mil heavier than all the rest of the archaeal lipids. So we can essentially draw a line at 110 per mil, and anything less than that is going to be the archaeal lipids. Quercetine is often quite often heavier of the archaeal lipids. So this is the value for archaea in the sediment. We also extracted the sulfate-reducing bacterial lipids and fatty acids, and we found that their values is -1 to -20. This means, first off, these values can only be derived from methane. They're clearly performing the anaerobic oxidation of methane, and since we don't really know much about the microbiology within these rocks, it's a great indication that they're probably playing some unknown role within the methane cycle. But then we started looking at the worms. Here is the delta 13C value for the bulk tissue from the worm. And its value is about the mean is hidden. It's right in the center here. These are the standard error colors. The mean was -91, plus or minus a few, which means it's also definitely getting its energy from methane. Uniquely, the lightest individual we got was -101, and this is a world record for the lightest isotopic ratio of a metazoan anywhere on the planet that we know of. So they're also definitely getting their energy from methane and methane cycling. So now we've decided to take a much closer look at the worm, because this is if anything's going to be eating archaea, it's going to be this worm. So in this figure, the left y-axis is the percent fatty acid, similar to what I showed you with the Yeti plant. We have all of the different fatty acids that were abundant either within the rock or within the worm on the x-axis. And then we have a secondary y-axis on the right-hand side, which is the isotopic ratio of the specific fatty acids from two individuals from two different rocks, both collected from this one methane seep off the coast. I then color-coded it with the knowledge of what we got from the lab. I should say hash-coded it from the knowledge we got from the lab. Every single thing that does not have the hashes through it, so for example, almost all of it, is what we found that worms of the same family can de novo synthesize within the lab. And so rather than looking at this blankly and saying, well, it seems to be having a lot of something that's making 18(1n-7), we can say it has a lot of things that it can de novo synthesize. There were two that we did not find in the lab, and that was a 16(1n-5 and an 18(1n-5. And if we look at the isotopic signatures of all of these fatty acids, it's, as we would expect, incredibly light methane derived. So let's compare that to what we found in the rock. So here we have, again, percent fatty acid on the y-axis, again, delta 13C of the specific fatty acid. And now I've color-coded the red as the sulfate-reducing bacterial biomarkers, which we know form part of that syntrophic partnership and perform this very important biogeochemical process. You can see that, as we would suspect, and actually we back this up with 16S phylogeny data for what was present within the rock, that the two most abundant belong to an AMI2 type association and sulfate-reducing bacterial partner. Its isotopic values were very negative, as you'd expect, if they're getting energy from methane. And this particular one lines up perfectly with 16(1n-5). Now, that's important for a couple reasons. There was only two, again, that we didn't find within our lab rat, and that was 16(1n-5, and the second was 18(1-5). But we also know what cellular machinery at least has, and that's what most metazoans have. And so we know that it can make a 16(1n-5 using the known cellular machinery to create an 18(1n-5. So essentially, this is a derivative of this that all comes from this diet. If you look at the other, essentially all the values for the isotopic composition match up and everything else. So it's a really great example that they're getting energy from the sulfate-reducing bacteria. Again, methane fuel habitat. But it actually was a bit more powerful than that. Because if you're trying to think of the impact this might have on a methane cycle or on the amount of rate processes, essentially going back to that lake and the phytoplankton example, it's going to have a much larger impact if it's digesting both the sulfate-reducing bacteria, which we learned from the previous slide, but also if it's digesting the archaea itself. And because we know that it's adding 2 carbons using its known machinery to the 16(1n-5, we can figure out what the isotopic composition of its food source is, because it's breaking its food source down to make those 2 carbons into acetate and add it to that 16(1n-5 to make the 18(1n-5. It's called lipid math. It's basic mass balances of carbon. And we come up with a value that its food source, what it's digesting and using to build its cellular machinery, has an isotopic value of -101 to about, or to -120. These are the 2 different individuals that we have. Now, if you plot that on all of the isotopic values that we have, this is just the more negative one, -128, it would appear that they're getting a large proportion of their energy actually from the archaeal lipids, because it's the only isotopic value which falls close to this, essentially the carbon value that they're consuming. So this is a great demonstration that not only are they eating the sulfate-reducing bacteria, they're also digesting and ingesting the archaea and using potentially even the archaeal lipids as a building block to build the sulfate reducer, to build their fatty acids. So essentially, are archaea a viable food source? Yes. Are any archaeal biomarkers incorporated in metazoan tissue? No. And does archivory actually occur? Yes, it appears that it does. So we think that the family Dorvalidae may be really used to this or adapted to this because of their jaws. They have dark chitinized jaws, and in fact, it's very similar to the radula of a limpet in the intertidal or a gastropod in the intertidal, because they can use it to scrape these archaeal aggregates off of rocks. But there's a lot of different there's a lot of different Dorvalidae within methane seep, not only in the rocks but also in the sediment, and each one of the species appear to have a unique isotopic signature. This is decades worth of data from my PhD advisor showing that each worm species, these are from Hydrate Ridge and Yale River, so off the coast of Oregon and Northern California. The specific species has its own kind of isotopic niche space that it occupies. And they also have different sediment horizons, so it looks like they're all able to eat different things. Now further, I took examples from all of these different species. They're color-coded to putative species here, and tried to find out what fatty acid or what sort of pattern may explain the isotopic signature relating to the amount of methane that they're consuming. By far, the best explanatory variable was the sum of 16(1n-5) and 18(1n-5, the same two fatty acids that we found within the rock of the animals that was eating the archaeal biome. Here's the scatter plot. That simple relationship explained 54% of the variance of that regression, which is pretty amazing. And even though this looks like these values are skewing this regression greatly, you can remove them, remove all the outliers, and the relationship stays essentially the same with no deviation in the slope. So steadily smaller. So what does that mean? Well, this may be the Darwin finches of the deep sea methane seep mud, because they each have different shaped jaws and different types of jaws, and at the same time have different diets, potentially providing different mechanisms to allow them to harvest archaeal aggregates to different extents. So I told you a story about symbiont-bearing Yeti crabs, and then also heterotrophic food webs involving archivorous annelids, showing first off that archaea are a viable food source, should be incorporated into food web models and trophic understandings around the globe. And then secondly, showing an example of a top-down force, potential top-down force on archaeal aggregates in methane seep sediments. Now, the extent of how important is this top-down force? This was the first step and showed that it actually exists. There is a trophic connection between the methane cycle and the different worms within the sediment and within this rock, but we don't know how important it is. And so that's going to be something that needs to be answered over time. But I started the talk saying we discover a lot of different, unique, novel things when we don't expect it. And this was another thing. If you park this up and just watch things for a second, you can see some pretty unique behaviors. So I want you to look at these two ones, which appear to have some sort of interaction. And these are both the same species. They're both Cubocuravida, just crabs have to molt like everything else and likely lose their bacterial mat when they do. And there's some sort of reaction or interaction going on here. You can see how they normally look. They just sit there and wave their arms back and forth, irrespective of anything else that's going on. But just about now, we'll see things change. I wasn't supposed to pause. Nor break. I'll tell a different story while this goes to that stage again. So you can also see that those bathymodial mussels, they tend to live within the authigenic carbonates. And again, pay attention to these. There's also a lot of, you can see some small vestimentiferous tube worms, and there's even almond-carrot shrimp. And one of the common questions we get is, well, are they actually eating shrimps or other things? We never saw them scavenging, and although the shrimps would get close to them, if they did, they would immediately get battered away. It was one of the few interactions we saw. And there's, within a small habitat, you find 60, 100 of every size Yeti crab. Here's an itty bitty little one. And they would go from just a few millimeters all the way up to about 4 to 5 centimeters in distance.
Was that a shrimp? Just that just off?
If it ran away,
yeah,
that would have been a shrimp. And also, if you look closely, you can actually see them use their third maxillary head to scrape off their bacteria periodically. So there's a question, is this similar to what we see in the intertidal? Is this some sort of competition of space? Because we know that there's only methane and seep fluid that's coming out in certain areas. Or is this some sort of other display? So we all know that crustaceans need to reproduce right after they molt. We know that that one had very little bacteria on it when it was essentially part of that display. So we don't know whether that was actually the passing of a spermatophore from the male to the female just after it was essentially molted. So we may have actually just watched reproduction, or we may have seen a competition for space display. We have no idea. So with that, I'd like to thank all the people that paid for this. If you invite me back in about 3 years, I'll tell you a story about how worms impact bacteria in the Antarctic. And many wonderful collaborators, many wonderful people at sea. And thank you for your time and attention.
Any questions?
Yeah. Epibiology.
just means living on biology. So one of the people like to argue about terms, and everybody was quite happy to say that bacteria are epibiotic on crustaceans, but nobody would agree that they were symbiotic. And so it's kind of splitting hairs, but you can call them epibiotic bacteria because they're growing on the exoskeleton of the crab. But I couldn't call it symbiotic in the literature because actually I couldn't figure it out because they agreed that I'd showed that they were eating and harvesting and increasing the productivity of the bacteria, but for some reason that wasn't allowed to be symbiosis. But so it's a way to avoid that literature snafu. Yes.
Is it also possible that since the ones that molt have little bacteria, could they be competing, trying to eat bacteria off of the more established crops?
So there's no reason why that's not the case, but I think what that may be instead is it could be them trying to get re-inoculated by the bacteria. And if you, I can go back, but there's one point where it essentially sticks its arm in the other one's maxilliped or mouth, and that may be them trying to get the bacteria off.